Okay, I'm in. Hello, Irene. I'm ready.
Hi, Peter. Go ahead.
Let's wait a minute or two.
Sounds good.
Dr. Grayson's available. Fabulous. I presume someone will turn the music off eventually. Irene, do you have a few announcements first? Someone keeps muting us. We can't hear you, Irene. If our tech person could just unmute the attendees, please.
Oh, there. Okay, there. Sorry about that. Okay, everybody. First of all, my name is Irene Tan. I'm the president of the Philadelphia Rheumatism Society. And I would like to take this opportunity to thank our corporate sponsors who are supporting the 2020 to 2021 Philadelphia Rheumatism Society virtual series. And our sponsors are AbbVie, Amgen, Aurinia, GSK, Janssen, Mallinckrodt, Novartis, and Sanofi Genzyme. Okay, now I would like to hand this over to Dr. Peter Merkel.
Well, good evening, everybody. It's good to see a good crowd. Welcome to the annual H. Ralph Schumacher memorial lecture. I can think of I can't think of anyone as good as Dr. Grayson to honor Dr. Schumacher, who is, Dr. Grayson, you're following several years of wonderful speakers, all of which who, in some way or another, honor Dr. Schumacher with their approach to science and medicine. And I think you share Dr. Schumacher's combination of passion for clinical care and actually looking at the patient in front of you and linking that to scientific questions. And I think tonight's lecture, Dr. Grayson, is going to really, I know who would have loved it is H. Ralph Schumacher would have loved this talk, because it really says, let's look at the patient, let's try to figure out what's going on, and let's merge the science using the most recent scientific methods, which is exactly what we're going to hear about tonight. Extremely exciting story about VEXAS. I'm really privileged to introduce Peter Grayson. Peter graduated from the Medical College of South Carolina from medical school, and he then came to Boston, where I first met him when we recruited him to Boston University Medical Center, where he was a resident in medicine and then a fellow in rheumatology, and did his advanced master's training there as well. Peter Grayson worked unfortunately with me, but he has been successful nonetheless, and he has gone on and he founded the vasculitis program at the National Institute for Arthritis, Musculoskeletal, and Skin Diseases, and in so doing continued the legacy of vasculitis at the NIH. And for all of us in rheumatology, those who've been around long enough know, there has been a long and storied tradition of vasculitis research and care at NIH, originally starting with Tony Fauci, the same Tony Fauci we see on the news every night, who did groundbreaking work in granulomatous with polyangiitis and Cohen-Wegener's, and Shelley Wolf, and then followed by Gary Hoffman, and then Carol Langford. And then there was a hiatus, and then Dr. Grayson moved to NIH and has allowed this program to flourish tremendously. As you'll hear, he's made it one of the world's leading centers for vasculitis care and research. And in particular, he has been groundbreaking in his work on imaging in large vessel vasculitis, as well as thinking about, from a translational standpoint, everything from how to think about the patient to the genetic underpinnings of vasculitis and vasculitis-like diseases. And I think this is an example of the story you're getting tonight of the kind of translational research. And people talk about bench to bedside, but I actually like to think about bedside to bench. Well, I think it's a bilateral, it goes back and forth, and you're going to hear that story tonight. So he is a wonderful speaker and award-winning educator, published over 80 articles in scientific literature. This is tonight's story is stemming from a paper in the New England Journal within the past 6 months that Dr. Grayson was the senior author on, as he helped lead the team that has gone to this thing. So I think you're going to find this an exciting journey. So I'm happy to introduce to Peter Grayson to give this year's Schumacher lecture.
Thank you very much, Dr. Merkel, for the introduction. I really appreciate it. Thank all of you guys for coming. And I first want to begin by just acknowledging Dr. Schumacher. And I understand that, you know, for many, many decades, he was a treasure within the Philadelphia rheumatology community and internationally. And I understand him to be just an excellent physician scientist, educator, quite prolific. His publication record speaks for itself. And the thing that I really respect about him in learning about his life is that even though he was so accomplished in medicine, he had a life outside of medicine and a lot of varied interests, including music and world-class gardening. And I definitely have a lot of respect for people that manage to find that work-life balance. So I apologize to my colleagues at the University of Pennsylvania for having to hear me give 2 lectures in a day. I apologize to my colleagues at Temple, who I think heard the VEXAS story from me a few weeks ago, although I've changed a little bit of it. But I don't apologize for the talk that I'm about to give, because I hope you find it quite exciting. Many of you are aware that this new disease was recently discovered called the VEXAS syndrome and published at the end of last year. But I'm here to tell you the backstory of the discovery and really to tell it to you from a very personal way, because I think it interweaves with my career direction. The story took a lot of twists and turns and unexpected, fortuitous events, and a lot of that was just being in the right place at the right time. And the theme that I want to talk to you about, beyond what is VEXAS, how do we recognize it, why is it important for rheumatologists to know about it, is to understand that this work required high-level team science. There were a lot of people involved, and there were both challenges and extreme benefits of working collaboratively in a disease like VEXAS. So one of the themes that I'd like for you to carry forward as I give this talk is the idea that working together across different disciplines with different stakeholders really is the way to move the needle in disease discovery programs. Okay, so the story of VEXAS for me begins at the end of 2012, and I was an assistant professor at Boston University, and my family and I decided to move to the DC area, and I pitched the National Institute of Arthritis and Musculoskeletal Skin Disease the idea that I would come and start a vasculitis program there. And as Dr. Merkel mentioned, there was a previous program that had ended in 2005. And so they graciously took me up on my offer, and I came and started working here at the Clinical Center, or Building 10, which is the largest research hospital in the world. And I remember arriving in 2012 and trying to think, what am I going to do? This is a place with amazing resources. I have a lot of questions I want to ask, but how do I match it to the environment? I need to be focused. I need to write an observational protocol, which will allow me to start bringing patients with vasculitis to the clinical center. And as I was trying to get everything in line, I got approached by a group at the NIH called the Undiagnosed Disease Program. And you may know this now, it's expanded into an undiagnosed disease network. But the UDP came up to me and they said, here's our pitch. We want you to serve as a consultant for our program. We think you'll find it very interesting because we see patients with vasculitis, and often we see patients with very atypical forms of vasculitis that just don't fit any classification scheme or any diagnosis. And we'd love to have your input. You probably only need to see a few of these patients a year. And even though I was trying to get focused and getting things up and running, I really couldn't pass up on that opportunity because the reason that I'd gotten into rheumatology was my love of the diagnostic dilemma, the medical mystery aspect of these complex multisystemic diseases. So I agreed to serve as a consultant without really knowing what I'd gotten myself into. And I'm going to share with you one of the first cases that I saw in the UDP. And this was a man in his late 50s who was previously healthy until 2011 when he developed a very strange constellation of systemic inflammatory symptoms, which included fever, orchitis, painful skin nodules on his extremities. He described these weird episodes of face swelling that I'll characterize further in a minute. He'd had 2 episodes of unprovoked DVT, and he mentioned at least 2 episodes of painful swelling of the external ear, sparing the lobule, that sounded a lot to us like auricular chondritis, but he didn't have photographic evidence of the events. And his doctors had really struggled to try to get his illness under control. They had given him various forms of medications, even at one point opting to give him cyclophosphamide, which was only temporizing. And the only thing that really helped him was high doses of glucocorticoids. But even then, he had ongoing inflammation. So he applied to the Undiagnosed Disease Program in 2013 and was accepted. And I went and saw him in our inpatient center at the clinical center with Amanda Umbrella, who's a rheumatologist that works in Dan Kastner's group at the fever team and specializes in autoinflammatory diseases. And we sat at the bedside with this patient, and he told us something really remarkable. He said that I'm on 25 milligrams of prednisone a day right now. If I drop my dose to 20 milligrams within a day, I will flare. And we thought, well, you're under observation, let's do that experiment. And so we did. We lowered his prednisone, and sure enough, within a day, he started to develop those painful skin nodules. That face swelling that he was telling us, this is a picture of it, it was mainly periorbital edema around his left eye. His C-reactive protein was 30 mg per liter when he was admitted and went up to over 300 in just a matter of days. And we did a biopsy of one of these skin nodules and saw medium vessel vasculitis in the deep dermis. And so putting together medium vessel vasculitis in skin with testicular pain and inflammation, our working diagnosis was atypical polyarteritis nodosa. And for the next years, we worked with his local doctors to manage his condition. And we tried a variety of medications, including biologic therapies, and nothing was very successful. He had a very profound skin reaction to anakinra. and he needed high doses of daily glucocorticoids. And then he started to develop some very bizarre hematologic abnormalities, which we would characterize as progressive bone marrow failure. He first had macrocytic anemia, and then he had progressive thrombocytopenia. And again, ongoing inflammation, multiple infections started to happen, which we thought were secondary to his immunosuppression. 2017, he started requiring blood transfusions due to his worsening anemia, and we did a bone marrow biopsy, and it was non-diagnostic. It showed hypercellular marrow with a myeloid predominance, and he had negative cytogenetics and genomic studies for myelodysplastic syndrome, which was our concern given the cytopenias and macrocytic anemia. So we really didn't know what he had. We still were operating under the assumption that he had atypical polyarteritis nodosa. We weren't sure what was going on with his bone marrow failure. We thought maybe it's just secondary to bad inflammation. And we soldiered on, and the patient got worse. So he began to, he continued to have fevers. He started to get these pulmonary infiltrates that were responsive only to high doses of intravenous glucocorticoids. He started to develop atypical infections. Now he needed platelet transfusions in addition to blood transfusions, ongoing inflammatory symptoms, multiple hospitalizations. And unfortunately, in 2020, he died from complications of his illness. And this is one of these cases that stay with you, in part because he didn't neatly fit one diagnosis. We thought he had polyarteritis nodosa, but there were so many things that didn't fit. And we were really impressed by just the severity of his inflammation and the treatment refractory nature of his disease, and this weird combination of hematologic problems with rheumatologic problems. And I carried that case around in my head until 3 years later, in 2016, I saw two more cases that were pretty stereotypically similar to this one. The first one was seen at the request of the UDP, and the second one, Amanda Umbrella pulled me into Fever Team Clinic, and we saw. And at this point, we took the two cases from 2016, not this original one that I told you about, and we sent them off for whole exome sequencing studies, thinking that because they're so clinically similar, maybe there is some shared novel genetic variant that's underlying this. We did the analysis of that data, and we didn't find anything. And we were stuck until 2019. In the summer of 2019, I got a phone call from a clinical geneticist working in Dan Kastner's group named David Beck. And David Beck was interested in the ubiquitin pathway, which is a fundamental pathway of cellular signaling and protein degradation. And David was interested in the ubiquitin pathway because many genetic diseases had been discovered with perturbations in ubiquitin-related genes. And David did something quite radical. He recognized that over time, the Undiagnosed Disease Program, Dan Kastner's Fever Clinic, they get exome sequencing studies on most patients that come through their program, yet only in a minority of them do they arrive at a genetic diagnosis. And so these genetic databases accumulate and build up. And so what he did is to take a genotype first approach, where he looked at their genetic data agnostic to clinical phenotype. So he had exome data in 2,560 individuals. He restricted his search to 841 genes related to ubiquination. He applied some filters that would ensure that the variants that he discovered were likely to be pathogenic. And lo and behold, a common novel variant never seen before in any human database popped out in 3 older men. And they had this mutation in UBA1 at codon 41. And these are chromatograms from these 3 patients. And when David looked at the clinical charts, he recognized that 2 of them were the patients I told you about on the last slide, and he gave me a call. And I was extremely excited to get that call because, as I mentioned, these are patients I'd thought a lot about. If you look at these chromatograms, the UBA1 gene is X-linked, and it is, these were men. So you should see single nucleotide peaks at codon 41. And instead, these men had a double peak indicated by the black arrows. So they had one reference read, and then they had one mutant peak. And so the first thing we set about doing was trying to figure out why we're seeing a double peak in a gene where there's only one allele in a man. And I remembered the first case, the one I told you about. And so I gave David DNA from him because he hadn't been sequenced. And lo and behold, he also had this double peak at codon 41. Now we have patient 4. And so we, after doing a couple genetic housekeeping analyses, we started to surmise that perhaps we are seeing a double peak because we're looking in blood, and there's different cell populations in blood. And maybe some of the cell populations have a mutation and some don't, and that's why we're seeing 2 reads. This is the idea of mosaicism, caused by a somatic mutation, a mutation that's occurring sometime later in life, not one that you're born with. And so to prove that, we did skin biopsies in these patients. And if you look at their skin fibroblasts, they're normal. They all have a single peak. There's none of that double peak with that mutant allele popping out. So now at this point, we realized we've got a somatic mutation in 4 men, all of whom had these weird inflammatory hematologic overlap syndrome. And it's probably in certain populations in blood and not in others. So we set about trying to figure out, where is it restricted in blood? And we took advantage of the fact that these patients were requiring bone marrow biopsies for their cytopenias to try to figure out what was going on with their marrow. So we were able to get cells from bone marrow as well as cells from circulation. And David Beck and another fellow at NIAMS named Keith Sikora began to purify cell populations in these patients. So I'm showing you chromatograms from one single patient at codon 41 of UBA1, and you'll see the red peak is the wild type, that's the normal read. The blue peak is the mutation. And if you look at these hematopoietic stem cells, the cells in your bone marrow that make all of your blood, you see equal proportion of red and blue. Some of these stem cells have the mutation and some don't. And when you chase that through myeloid lineages and end up in circulating mature neutrophils and monocytes, it's almost all blue peak, very little red, meaning that these myeloid cell populations are almost all mutant. Whereas if you go through the lymphoid progenitor line into mature T cells and B cells, you see a single peak, it's only red, so it's wild type. So none of the lymphocytes harbor this mutation. It's a mutation that is happening at the level of the stem cell that's then lineage restricted to myeloid cells. And this is just showing that quantitatively by the variant allele fraction. So in skin fibroblasts, no mutation. T cells and B cells in circulation, no mutation, but a really high allele burden, a high clonal population in myeloid cells. We went and got together as a group, and we started looking at the bone marrow biopsies and aspirates from these patients. And these are aspirates from the first 3 patients that we had genetically identified. And we sat down with Kathy Calvo, who's a hematopathologist at the NIH, and we were shocked at what we saw because it just jumped out at us. In all 3 of these patients, in the erythroid and myeloid precursor cells in the marrow aspirates, we saw these little white dots, these vacuoles in the cytoplasm of the cells. And to date, every patient that we've seen with this mutation has these vacuoles. It's a real diagnostic clue for the disease. But at the time, we just had these first 3. Dr. Calvo went away and said, I'm going to go look through the records in NIH and see if I can find more patients that have these vacuoles in their marrow. I haven't seen this in many conditions. The only ones I can think about is copper deficiency can do this, and these patients weren't copper deficient, we checked. Severe malnourishment, alcoholism can do this, which didn't apply. She said, these are just unusual things that we don't see very often. So she went away to think about it. And we set out to figure out, okay, we have a mutation in UBA1 in myeloid cells in patients with really bad inflammatory diseases. What is that mutation doing to the myeloid cells? And this is electron microscopy from a bone marrow derived monocyte from a healthy subject. And you can see a nice looking cell. This is a bone marrow derived monocyte from a patient with one of these UBA1 mutations. And you can see it's extremely unhealthy because the mutation is toxic to these cells. And Keith Sikora in Bob Colbert's group sorted different blood populations, so neutrophils, monocytes, and then the lymphocyte populations in blue. And he did bulk RNA sequencing. And what we saw in the neutrophil and monocyte lineages, so in the myeloid lineages, there was upregulation of a ton of different innate inflammatory cytokine pathways, including IL-6, IL-1, TNF-alpha, interferon gamma, interferon alpha. It was a state of hyperinflammation in these myeloid cells, which likely explains why they were so treatment refractory. So I'm going to give you a little crash course in ubiquitin. I didn't know much about this before we started the project, but Akeem Warner is an expert in ubiquitulations at the NIH, so we got him involved. He's a co-senior on the manuscript. And the short of it is that ubiquitin is very essential to cellular homeostasis. As I mentioned, it can affect cell signaling, it can affect degradation of misfolded proteins. And it's a three-step process. There is E1 enzymes, E2 enzymes, and E3 enzymes. And at the level of E3, there's over 600 enzymes. There's a lot of specificity here. And this is where a lot of genetic diseases have been discovered, which is why David initially was really interested in the ubiquitin pathway. If you go back though, E2, there's not as much specificity. There's only about 40 enzymes. But E1, the master switch to all of cellular ubiquitination, is controlled largely by two enzymes, UBA1 and UBA6, and UBA1 does about 99% of it. So we're finding novel mutations in men in UBA1, the master switch that controls all cellular ubiquitination, which is why we think this mutation is somatic and restricted to blood, because if it were in the germline, it would be lethal. A little bit about UBA1 genetics. So there's two protein isoforms from the UBA1 gene that are translated at different start sites, defined by a methionine at position 1, which translates UBA1A, the long version of UBA1. This localizes to the nucleus, is essential for cell function. And then there's a shorter isoform, UBA1B. And curiously, the translational start site for UBA1B is methionine at position 41, which is exactly where we're seeing all of these mutations in our patient population. I will point out also that there is one germline mutation in UBA1 that's associated with a disease. It's X-linked spinal muscular atrophy, a mutation site more distal in the gene, perhaps not having such a deleterious effect on function of the gene. But here we're seeing a mutation at codon 41, and we thought then, okay, this is going to affect translation of this shorter isoform. Perhaps it won't be translated. And this is the isoform that goes to the cytoplasm, and that's why they're getting impairment in ubiquitination and these forming these toxic vacuoles in the cell and causing hyperinflammation. The story was actually a little bit more complicated than that, and I'll take you through this quickly. This is cell transfection experiments that David Beck did. Here is normal UBA1A, 1B. When we introduce these mutations at codon 41, you lose UBA1B and a smaller band emerges, which we're calling UBA1C, which we think starts at the next available methionine, which is position 67. So when you alter position 67, you lose this third band. So we're seeing in the patients this emergence of UBA1C due to these mutations at the start site for translation of UBA1B. And when we do these things called thioesterification assays, which looks at the function of that enzyme, we see that UBA1C is catalytically impaired, so it does not initiate ubiquitination. So at this point, we have this story that we have 4 men, they have atypical forms of vasculitis, they have somatic mutation in a novel gene, codon 41 of UBA1, they lose cellular ubiquitination, they get myeloid inflammation. We think we have a really cool story. We're ready to publish this. But the story took an interesting twist. And to understand this, I have to backtrack a little bit and explain my career direction. So as Dr. Merkel mentioned, I came to the NIH to study many different forms of systemic vasculitis. And while I was there, one of the fellows in the NIAMS program named Marcella Ferrata approached me because she wanted to do research in relapsing polychondritis, which is a rare autoimmune disease where you have cartilage inflammation. About 10 to 20% of patients with RP can have vasculitis, and it can look very similar to granulomatosis with polyangiitis. So I thought it was a clinical fit for me to study this disease with her. And more importantly, I really liked Dr. Ferrata, and I thought she was going to be a wonderful investigator. So I agreed to start studying this disease with her. And over a couple of years, we started seeing cases of relapsing polychondritis. And what you'll notice when you start seeing these patients is there's profound clinical heterogeneity, just like there is in many systemic rheumatologic diseases. And our group and others have used data partitioning methods to understand that heterogeneity. And this is a study from France that we really like. In 142 patients, they did cluster analysis based off of clinical symptoms, and they saw this constellation of symptoms in about 9% of their cohort, which they labeled hematologic relapsing polychondritis. And you look at the characteristics of this subset, they are older men, they have thrombosis, they have skin disease, they have an increased risk for death, and they get myelodysplastic-like syndrome. So again, severe inflammation with hematologic problems, which sounded a lot like our cases where we were seeing the UBA1 mutations. And as you may recall from the first case I showed you, that patient did tell us, although we didn't really quite pick up on it at the time, that they had auricular chondritis. So we started looking in our cohort for patients with this hematologic RP phenotype. And here's one of them. This was a 69-year-old man. He came in with fevers, a lot of skin complaints, and I'll show you some of them. On exam, he had synovitis in his hands and wrists. The bottom picture is these plaque-like lesions on his back, which were biopsied and showed neutrophilic dermatosis consistent with Sweet syndrome. The top picture is his ear and shows auricular chondritis, hence his diagnosis of relapsing polychondritis. He had pretty bad disease with elevated acute phase reactants, and we wanted to go back and look at his hematologic problems. So when we looked at that more closely, we realized that this guy had macrocytic anemia that was progressive. He had thrombocytopenia, and we went and pulled his marrow aspirate and looked at it again. And lo and behold, he had those characteristic vacuoles that were so specific to those patients that we had seen with the UBA1 mutations. So we had DNA on our cohort, and we sequenced UBA1 in 92 patients with relapsing polychondritis in our cohort. And we found that 7 of them, or 8% of the cohort, had these novel mutations at codon 41 in UBA1. And all of these 7 patients were older men with this hematologic RP phenotype. So at this point, we realized that UBA1 was defining a new disease syndrome. Initially, we'd seen these 3 patients identified by this genotype first approach, and then we identified additional patients based on the presence of bone marrow vacuoles. Dr. Calvo came back with a list of patients that had these vacuoles, including a patient in Dan Kastner's cohort whom he had not sequenced. And we went and pulled his DNA, sequenced it, and he had a UBA1 mutation. We also started to find more patients, looking for patients with this combination of inflammation and progressive bone marrow failure. And to date, we have genetically confirmed over 60 cases of this condition, all of them with somatic mutations in codon 41 of UBA1. And we named this syndrome VEXUS, which stands for vacuoles. As I mentioned, those really characteristic bone marrow aspirate finding. E1 ubiquitin activating enzyme, which is UBA1. It's X-linked gene. It produces autoinflammation in myeloid cells, and it's a somatic mutation. It's not germline. And here you see the salient clinical features: inflammation in the lungs, the skin, the cartilage, and bone marrow pathology. We described the first 25 patients in a New England Journal publication, and one thing you'll note is that they're older men. The median age of disease onset was 64, and this disease began exclusively in the 5th decade of life or later. And all of these were men because, as I mentioned, this is an X-linked mutation that likely functions as an X-linked recessive disease. So women are protected by their second allele. What got us really interested as rheumatologists is that these patients get a lot of inflammation, and it can look like a lot of rheumatologic diseases. We applied existing diagnostic criteria or classification criteria to call this diagnosis for each of these patients, but 60% of the cohort had relapsing polychondritis. Many of them had Sweet syndrome, biopsy-proven Sweet syndrome. Many of them would evolve over time into overt myelodysplastic syndrome that met WHO criteria for that. Similarly, a smaller subset evolved into multiple myeloma. I told you about the polyarteritis nodosa phenotype, and we even found one patient with biopsy-proven giant cell arteritis. A little bit about the phenotype. The chondritis is limited to the ear and the nose in our experience, although this may be changing. In Japan, it seems there may be airway involvement in VEXUS syndrome. But what we see is pretty mild chondritis. These patients do not have damaging issues related to inflammation. They don't get saddle nose deformity. They don't get cauliflower ear. They get subtle chondritis that comes and goes. They have a range of skin manifestations, so I think dermatologists need to be aware of the condition. Anakinra universally causes severe skin reactions to them. We avoid that medication, which are what these two pictures are showing. They have that very curious periorbital edema finding. They get different versions of vasculitis. This is leukocytoclastic vasculitis, palpable purpura in the foot of one of our patients. They can get the deeper dermal medium vessel vasculitis. And this is a picture of neutrophilic dermatosis. They also get pulmonary involvement. They get this transient infiltrates in the lungs that require high doses of intravenous glucocorticoids, or they get sick quickly and end up intubated and can die from this. We actually did an open lung biopsy on one of these patients, which is shown here. It's neutrophilic alveolitis. It's a sterile alveolitis, probably from these mutant neutrophils. And on that same biopsy, we actually saw evidence of bronchial arteritis. So vasculitis of a bronchial artery. I've never seen that before in my career. These are common manifestations of the disease. It tends to start with macrocytic anemia and fevers. And you can see there's a very high thromboembolic signal. About 40% of our cohort had a DVT or PE. This is a cartoon for a upcoming publication summarizing the disease. And really the way I think about this disease is that it's a combination of an inflammatory disease and a hematologic disease. And you can see they get multi-system organ problems that can lead to a variety of rheumatologic and hematologic diagnoses. So this is our Vexus man, the older man. We put in some wrinkles. The challenging part about the disease is it's difficult to treat. And we've tried a number of steroid-sparing therapies, which are listed here, none of which were particularly efficacious. The only thing that really kept the disease under control was escalating doses of daily glucocorticoids. And as a result, it's a progressively morbid condition. Mortality in our cohort has been over 50%. There's a lot of complications, mechanical ventilation from the neutrophilic alveolitis. They get transfusion-dependent anemia, all the side effects of chronic glucocorticoid use, and they can evolve into hematologic malignancies. Because it's been so treatment refractory, we are now considering more aggressive therapeutic measures. We know of 5 patients around the world with Vexus syndrome who have been successfully cured of disease, we think, from allogeneic bone marrow transplantation. So because it's a disease of bone marrow, we think that bone marrow transplantation could be curative, and we've written a research protocol, and we're going to start doing that in selected cases at the NIH. And ultimately, one of our aspirations is to do gene editing therapies. This is a situation kind of like sickle cell disease, where the mutation is confined for the most part to one amino acid. So in the future, we think we could actually treat this by gene editing the patient's own stem cells. This is the resultant publication. It came out on New Year's Eve last year. I put this up here because there's over 60 co-authors. It truly was an effort of team science, with hematologists, geneticists, rheumatologists, immunologists, all working hand in hand to solve a medical mystery. I want to highlight 3 implications from the work. The first one is that somatic mutations are causal in rheumatologic diseases of adulthood. I'll talk about each of these in a second. The second one to discuss is the genotype first approach. And then the third one is I will make the argument that Vexus is a prototype for a new class of diseases. So there's a very nice editorial that in the New England Journal accompanying the article about somatic mutations and diseases. And somatic mutations occur after conception. And we are, I think, really comfortable in rheumatology thinking about germline or mutations that we inherit from our parents as being disease causal, looking for mutations in early onset cases or in cases where many family members have it. This is a totally different paradigm. It's a different way to think about genetics. Some of the early cases that suggested this were where you saw clustering of rare autoinflammatory diseases in children and siblings, but the parents didn't possess the mutation that the children had until you actually test gonadal tissue from the parents. So looking for mutations that were acquired in sperm in the father, so that it's not in any other cell, but it's passed along to the children. And if you see acquired mutations at embryogenesis and fetal development, you can get mosaicism. Some of the cells in the patient are mutant and some of them are wild type. But Vexus is out here. It's a disease of adulthood. It's exactly like the cancer, the neoplasia model. And we're really comfortable thinking that a mutation in a cell that provides a proliferative growth advantage is what a cancer is. And this is a different concept, but it's the same thing. It's a mutation in stem cells that's causing that cell to clonally expand in blood, and all of the blood that stem cell produces is pro-inflammatory blood. I think this is going to be a mechanism in many rheumatologic diseases beyond just VEXAS, and there is emerging literature to support this in many different diseases. So this is really cool stuff to check out. If you haven't read clonal hematopoiesis literature, it's really interesting. This disease was discovered by a genotype first approach. Traditionally, we rely on astute clinicians recognizing stereotypical presentations at the bedside, or getting lucky with these cases of familial aggregation where you can sequence many family members and find a gene that's causing a disease relatively expeditiously. But VEXAS turns that on its head, and it actually starts with the genome as we're aggregating all of these data over time, and then looks at the phenotype secondarily. And I think it's this approach that allowed us to see a disease that was kind of tricky to spot, and at least initially. But ultimately, it's the interplay between the two that matter. And what allowed us to expand the phenotype and understand the disease was our interaction of looking at the genomics and really deeply phenotyping these patients clinically. As I mentioned, I think VEXAS is a prototype for a new class of disease, and I'm trying to throw this term out to see if it sticks. So you have autoimmune diseases, and you have autoinflammatory diseases, and I think VEXAS is a hematoinflammatory disease, which I would define as a somatic mutation in blood causing hematologic mosaicism with systemic inflammation and the tendency to evolve into a myeloproliferative, myelodysplastic, or lymphoproliferative disease. You can't have a class of diseases if you just have one, but I think there are several examples of a hematoinflammatory disease in rheumatology. One of them is Erdheim-Chester disease. This is somatic mutations restricted to histiocytes in the BRAF gene in about 50% of these patients. So they have hematologic mosaicism. They have systemic inflammation, and we're interested in these patients because they get really profound aortitis. And they can, over time, evolve and develop myeloid neoplasia. I think there's evidence of this in Sjogren's syndrome with the connection between cryoglobulonemic vasculitis and lymphoma. I think there is evidence of this in Felty syndrome with large granular lymphoma. And I think that we're going to discover a lot of diseases that are in this paradigm in genes other than UBA1. We have known for a while that autoimmune diseases cross-interact with myelodysplastic syndromes, and there's a lot of people that have written about this clinically. These articles were really important to us as we're thinking about VEXAS. And here's table 2 from one of that second article that I showed you, looking at the types of autoimmune diseases that are associated with MDS, or chronic myelomonocytic leukemia. And here you see the usual suspects: relapsing polychondritis, PAN, GCA, Sweet syndrome. We think VEXAS is probably explaining a lot of this association, but not all of it. And if you look, there are a bunch of other diseases that are represented. These we think are really good cases to do deep sequencing, screen them for VEXAS with UBA1, and if not, keep sequencing the rest of the genome. So how do you spot VEXAS? It's really quite easy. And we did a simple clinical algorithm in our RP cohort that we just published in A&R. And it goes like this. If you have a patient with ear or nose chondritis, you can predict with 97% accuracy if they have VEXAS syndrome, if they have a mutation in UBA1 based on 3 clinical variables: patient sex, the MCV, and the platelet count. And so if the patient is male with an MCV of greater than 100, it's highly likely that they have VEXAS. If their MCV is less than 100, but their platelets are less than 200, it's highly likely they have VEXAS. And I think you could probably substitute other things, like if a patient has medium vessel vasculitis of skin, male, MCV, platelet counts. I think these are going to help us really quickly and easily pick off these patients. One of the phenotypes, and the first phenotype we saw, was PAN. So a little bit about how VEXAS fits in the historical context of PAN. It's an interesting history, and I took a lot of this from a very nice review article that Eric Madison published. The first case of PAN pathologically was described in 1852, where you're seeing these microaneurysms in the stomach of a young man. 1866, Kuzmal and Meyer described what they called periarteritis nodosa. The nosology of disease has evolved over time. In 1908, we landed at polyarteritis nodosa. And then for the next 100 years, we have been subsetting this disease. So in 1951, we recognized cutaneous limited PAN. In 1970, hepatitis B was identified as a causal factor in many patients with PAN. In 1990, there was ACR classification criteria for PAN, which were almost obsolete 2 years later, because we then recognized that many of those patients actually had ANCA-associated vasculitis in the form of microscopic polyangiitis, so subsetted out that away from PAN. In 2014, Dan Kastner's group and a group in Israel described childhood onset PAN due to mutations in adenosine deaminase 2, or the syndrome known as deficiency of adenosine deaminase 2, DADA2. And I think VEXAS fits into that history. This is yet another subset happening within PAN. And I think given the nature of polyarteritis nodosa, we're gonna see further subsets of disease come out of patients with this clinical diagnosis. But a pearl, polyarteritis nodosa spares the lungs, VEXAS does not. We had one patient with biopsy-proven giant cell arteritis. Now we've learned of a few more. In the case that we saw, this was a patient who had rapidly progressive bone marrow failure and died within a year of diagnosis. So he was typical GCA in the sense that he had biopsy-proven disease. He was atypical in the sense that he had this progressive bone marrow failure. Many of our patients with VEXAS are older men. They develop headaches non-specifically. They have very high acute phase reactants. They can even, because of their thromboembolic signal, they can get episodes of amaurosis fugax and stroke. So a lot of times people are entertaining the diagnosis of GCA. So what happens if you deep sequence just classic cases of GCA? So in our cohort, we sequenced that one patient up front because we thought clinically they had VEXAS, but then we took 47 more who just had more typical GCA, and we sequenced them looking for mutations in UBA1. What percentage of GCA is VEXAS going to be? And we didn't find any additional cases in those 47, only the patient that had the clinical phenotype. So at most 2%, but probably less. So I think patients with VEXAS can be diagnosed as GCA based on histology or clinical features, but it's unlikely that there's a high prevalence of VEXAS within GCA. You really want to be looking for these patients that have accompanying bone marrow failure. This is what happens to the natural history of the disease in terms of the hematologic aspects. So this is a single patient. They had inflammatory disease as the onset. That's usually the pattern. That's why they're recognized in rheumatologic clinics. And macrocytic anemia that over 5 years after symptom onset became transfusion dependent, and progressive thrombocytopenia. These green arrows are bone marrow biopsies that happen serially, all of which showed hypercellular marrow and all of which showed those abnormal myeloid cells with vacuoles. But eventually, 6 years after disease onset, this patient developed genomic evidence of myelodysplastic syndrome. So this patient did convert into what was considered more typically MDS. We are starting to get a sense of the prevalence of VEXAS, and it's quite interesting. So we actually use the Isinger database, which is a population database, and we found 5 cases of VEXAS syndrome out of 120,000. That's a disease prevalence of 1 in 30,000. To put that in context, that is similar to the prevalence of ANCA-associated vasculitis. And while that may be an overestimation, I still think this is not going to be an ultra rare disease. For example, it looks like this disease is at least 3 to 5 times more common than a disease like Takayasu's arteritis. It's just being dispersed among different care providers. Go into an MDS clinic, you're going to find patients with VEXAS syndrome. Go into dermatology clinics, you may find patients with VEXAS syndrome. And I think many of us as rheumatologists have seen these patients. The last thing I want to end is kind of the lessons in team science from the project. The first thing I learned is kind of a no-brainer, but was important to me, is that genomic data is powerful. Ethics aside, I think it's important to get sequencing data on patients and combine that with good clinical phenotype data. We see these 1 in a million cases all around the country. We have these insane cases that don't quite fit the textbook. These are cases that we should be sequencing, and we should be working together to try to identify molecular underpinnings of disease. I learned that hematologists and rheumatologists don't speak the same language. I learned this from the patients, because the patient's experience is they would go to the rheumatologist, they had bad inflammation, the rheumatologist would struggle to try to get that inflammation under control, but would say, you've got progressive bone marrow failure, there's something going on in your marrow that may be driving this, I don't know what it is, go see your hematologist. The hematologist would do bone marrow biopsies. They wouldn't meet any established criteria for a disease that they were aware of, and they would fall back on saying, this is due to inflammation. Go back to your rheumatologist, get the inflammation under control, and back and forth these patients would go. And I think this discovery gives hematologists and rheumatologists a framework to start working together more collaboratively on these patients. The other thing I realized as a rheumatologist is that hematologists and oncologists are very good at incorporating clinical genomics into practice. They are very comfortable with the idea of somatic mutations. They're very comfortable with profiling tissue and making therapeutic decisions based on molecular characterization of disease. We don't do this as much in rheumatology. And sometimes we even fall back on the concept that we are so comfortable in dealing with uncertainty that we can make decisions in the face of uncertainty. And we perhaps forget that it is possible to boil these complex phenotypes down to their essential elements. We should be striving to incorporate genomic medicine into our clinical practice. I learned, this is the most profound thing I learned, is that discovery is a process and not an event. There were a lot of things that happened along the way that were really important observations. When David did the genotype first approach and found the 3 men with the shared mutation in UBA1, that was an amazing observation that really set things forward. When we realized that these were in patients with relapsing polychondritis, and then Sweet syndrome, and then giant cell arteritis, and the phenotype evolved, when we realized the bone marrow vacuoles and we could use that to find other patients, these were amazing events. But true discovery took 1.5 years of 60+ people working hand in hand to try to unravel what was happening and to understand this disease in all of its pleomorphic nature. And ultimately, even though, as you can imagine, it was a bit like herding cats at times, trying to get everybody working towards a common goal, and everybody had their own individual interest and were progressing the project in their own ways, I think that this discovery is really a testament to team science, and it's a testimony to the best parts of the NIH intramural program, where we had the time and resources to work together across different disciplines to understand and characterize this disease. So with that, I thank people in the vasculitis program at NIAMS, some of the key people on Team Vexus, and you for your attention. I'm happy to take any questions. Thanks.
Dr. Grayson, that was a brilliant talk. How wonderful to see the unfolding of discovery of this disorder. So I have a patient not similar to this disease, but I just saw a patient with lupus. She's in her 40s, so different gender altogether, but systemic lupus with lupus nephritis and a very curious neurological disorder, axonal neuropathy, weakness, can't walk. And she's been transfusion dependent. I have to go back to see what kind of anemia, but it's not macrocytic, I don't think. But bone marrow is hypercellular, and we can figure out there's not much of hemolysis peripherally. So it's been very curious, but I think I'm going to ask them to look at the local vacuoles on the bone marrow biopsy. I don't know what I'm dealing with at this point. It's a patient I share with Temple team, so maybe I'll pick their brain after the talk also. Okay, well, open to question, and I don't see anyone raising their hands yet, so I'm going to give a few moments. Wait, I'm reading something here. Let me see.
Yeah, I see one in the chat. Is there something about UBA1 that provides a selective advantage to these cells? Is there a proliferative advantage even prior to the onset of overt myeloproliferative disease, MDS? So this is the fundamental paradox of the disease that we don't understand. You know, the mutation is not tolerated in lymphoid cells. It's selected out in the marrow so that in circulation, all of those T cells and B cells, they don't have the mutation. And it's likely because B cells are really intolerant to perturbations in ubiquination. They can't tolerate misfolded protein, whereas myeloid cells may not necessarily be as profoundly affected by defects in ubiquination. But we don't know why does a toxic mutation to myeloid cells allow for clonal expansion. And if you work with myeloid cells from these patients in the lab, they're very difficult to work with. They die of many different forms of cell death. And So something is happening at the level of the marrow where either it's giving them a selective advantage, or at least it's allowing them to survive perhaps in harsh inflammatory conditions, and that's why they expand. We're not sure, but I think the story has something to teach us about ubiquitization and hematologic maturation. And if we understood that paradox, perhaps we could treat the disease.
Okay, looks like from Blake Fishman, Dr. Schumacher would have loved your talk. From concern for a take-home patient to utilizing the newest medical technology to using a team to solving a puzzle. Thank you, great talk. That's a comment. From Quasar Huzef. If we find vexus with vacuolar myeloblast, then how should we treat this? Should we consider stem cell transplant in those?
It's a tricky thing, like which patients are you going to offer stem cell transplant to, and when do you do it in the course of disease? This is what we are struggling with. It's a disease of older men, so as you can imagine, many of them are not good transplant candidates due to functional status, comorbidities. And if you wait too long with the disease, they will inevitably develop comorbidities from chronic glucocorticoid use and from severe inflammatory disease that would make them poor transplant candidates. So we need to identify the risk factors for severe disease progression so that we can target specific patients earlier on in the course of disease. We think we've got that a bit worked out. So I don't think allogenic bone marrow transplant is going to be for everyone. It's probably going to be for a minority of people. Therefore, we need to understand what's happening at the cellular level and develop targeted treatments for this disease, because most patients, I think, are going to only be utilizing medical therapy as their hope for treatment. But it's hard because you get in these situations where you may transplant someone who's relatively healthy and there's risks of transplant.
Thank you. Next question from Peter A. Are you seeing a broadening of the phenotype with more cases, specifically from a mortality or speed to death point of view?
We have not seen a tremendous broadening of the phenotype, which we expect to happen. And we are really happy that it hasn't broadened too much, because I think it's a testament to how much effort we put in phenotypic characterization. We looked pretty carefully at that, and we tried to consider different diseases in which this disease could be hiding. There are many candidate diseases. I think it's probably hiding out in Behçet's disease. It's probably hiding out in antiphospholipid antibody syndrome, but we haven't seen cases like that. If you look at the literature in the last couple months, there's a bit of a mini explosion of Vexus papers from around the world. People are discovering this in Asia and in Europe, and the phenotype is broadening a little bit, but it's really still that core element of inflammation of skin, inflammation of cartilage, inflammation of lung. It still seems like the chondritis phenotype is the most common. I didn't go into it a lot, but if you look at the mutation that happens at codon 41, a methionine can either become a valine, a leucine, or a threonine. And depending on which one it becomes, we actually think that's associated with speed, with mortality rate, and with symptoms. So we do think there is a genotype-phenotype correlation in the disease.
Yes, one of the things I was wondering when I asked it is, first of all, a fantastic talk, and I do agree that Ralph would have loved this, and he would have gone back to the lab to look for new mutations and called you at any hour. So in terms of speed of death, I mean, I guess the question is, there are probably these cases hanging out in people's clinic and practices, but you don't necessarily maybe even think about it till they get quite sick. So do you think, you've talked about people, they sort of get to your attention because they get quite sick. You talked about the screening level, but I guess I'm wondering if that range, what's that range between when there's the first clinical sign, which could be obviously asymptomatic, and explosion?
The median survival time in the cohort, and this is true for the first 25, it's held true as we now have over 60 cases, and we know of over 200 cases in the world, has been 10 years from symptom onset. So 50% of these patients will die. What's
the symptom? Is it the rheumatologic symptom?
Usually the first symptoms are fever or rash or the cartilage problems. And then the first lab symptom is macrocytosis, which over time evolves into macrocytic anemia. So I would say, as rheumatologists, we need to be paying attention to the MCV. It's not just the drugs that are causing elevations. If you see an elevated MCV in a patient that's presenting with a systemic inflammatory disease, it should trigger a little bell.
Next question from Sally Holman Moore. How do we send you patients? Do you want just the blood or something else, or for the patients to go to NIH?
That's a very nice question. Thank you for asking. So David Beck's lab at the NIH has CLIA certification to do this as a diagnostic test. It lacks an ICD-10 code now, I guess, but we can test these patients for clinical purposes. We don't need research consent to do it. You can get in touch with me and we will tell you how to do it. Often we will, either the lab at your institution can draw the blood and just directly send it to us. We will test them and issue a report to you. Or sometimes if the patients can't get to your lab, we can consent them directly over the phone and do it through a research mechanism and mail them a test kit. I think there are centers around the country and world that are developing testing capabilities for this. It's actually not that difficult to screen them. And I think commercial companies like GeneDx will likely be doing this pretty soon. But in the interim, until testing is more universally available, we are happy to test your patients for free at the NIH. If they don't have a mutation in UBA1, then but they have an interesting phenotype of hematologic inflammatory overlap, then we would consent them for a research study where we're doing whole exome sequencing or whole genome sequencing at deep coverage, looking for novel mutations in other genes.
I'm going to jump in for one sec and let people know if you don't know, the University of Pennsylvania is actually now a site for the Undiagnosed Diseases Program, because the NIH can just, they've spread it out across the country because there are many, many more applications. So not for VEXs, although we're happy to see that because we have a relapsing polychondritis program that we work with Dr. Grayson's group on, but that Dr. Banerjee is running. But for those, like what you mentioned, Irene, if you have an undiagnosed problem, the patient has to apply through the portal, and then you have to provide a letter. Again, only some of the people are accepted, but I think we're going to be a source, that is we rheumatology, of new mutation-driven diseases that will be seen through this type of technology, because this program funded by NIH has the capacity to do whole genome, whole exome and expression work, which you can't just get done commercially. So not necessarily for the VEXs, but for these other ones that I think you would agree, Peter, there's almost certainly many diseases hiding in plain sight in our clinics.
Peter, so is it searchable? If I refer a patient to a Penn website, is it searchable on diagnosed program? Okay, you just went mute, can't hear you.
There's an Undiagnosed Disease Network website, and you can get to Penn through that.
Correct. You just look up Undiagnosed Disease Network. You don't actually apply to Penn, you apply to the program. And for patients that live in Pennsylvania or New Jersey, they'll be referred for screening. It's a complicated and slow process, but it does happen. And I'm on the evaluation committee, and we've seen some of the people in the audience who have actually sent patients, but it's only a few get in.
But we have some more questions. Art Hopper, since steroids work and the course is downhill, has there been consideration for IVIG?
We've tried it with limited success. I mean, most drugs that we try in this disease have partial efficacy that's usually transient. Our preferred biologic is tocilizumab. There's a pretty high IL-6 signature in the disease. But what our experience is you give them tocilizumab, they clinically improve for a short period of time. Sometimes their blood counts even improve, but eventually the disease progresses through that. There is a case report, I think, of a single patient of excess treated somewhat successfully with IVIG, but I don't think that that's the curative gun we're looking for. There is a signal of hypomethylating agents, so drugs like azacitidine, where it looks like that actually may impact disease and reduce clonal burden of the mutation. That right now is only being given in the ones that develop overt MDS with mutations that suggest problems in methylation. But it is a drug that we're thinking about perhaps instituting earlier in the disease course. Yeah, and then earlier to the earlier question, we can diagnose your patient with mail-in blood samples, but we also would love to have them come to the NIH, particularly once the world opens up a little more, particularly ones that may be candidates for transplant. That could potentially happen here.
Okay, one question from Nihal. I'm going to destroy your last name, so I won't pronounce it. Have you looked for the production of UBA1C in other patients with similar phenotypes that lack the MET41 valve threonine leucine?
Yeah, it's a good question. So if we just screen them for mutations at codon 41, are we going to miss patients that have VEXA syndrome with problems that still affect UBA1 isoforms? And the answer is that we've discovered 2 novel variants that have been reported by various groups. There's a splice junction motif at intron 2 and 3, so it knocks out UBA1. That's been reported by a few groups. And there is one patient that has a novel variant at codon 56, I believe, that also affects UBA1B, the shorter isoform, in a temperature-dependent manner. We are not systematically screening these patients by Western. We're screening them by deep sequencing of the gene itself. But yeah, I mean, you start to accumulate enough patients that have the exact clinical phenotype, but they lack the genotype. Then you start thinking about ways that maybe we're missing things. Maybe it's not just point mutations that are picking up by sequencing. Maybe there's something else going on that we need to do other approaches to see. So these are considerations for sure.
Dr. Georgka has a patient. Can you see the chat, Peter? Have you looked for the production of UBA?
I just answered that one. Yeah. In a roundabout, long-winded way.
Yeah, it's just, but are you looking for, do you think there'll be promoters? Do you think there'll be other facets of this, or you just haven't found that yet?
We found 2 new variants, like I mentioned, but 99% of these patients have mutations only at codon 41. And that codon is very specific because it initiates translation of the shorter isoform. I think if there's too much problem with the gene, because you remember, like, this is a fundamental gene to all cell function. So, you know, it can easily be lethal. So I do think that we're going to see a restricted, smaller set of variants, unlike some diseases where there's mutations throughout a gene. Loss of function mutations throughout a gene can cause, like DATA2, for example, there's a ton of different variants that define DATA2. I think VEXA is going to be much more restrictive because of how the mechanism of how it works.
Hey, Peter, what was the I'm just looking for. Well, how old is the youngest patient you have in your cohort?
The youngest patient is 45 at the onset of disease. And we are hearing of cases that are a bit younger, but they never, when we sequence them, they never end up actually having UBA1 mutations. We also have heard a little bit of females that may have VEXA. There was a report out of Japan of a woman who had VEXA with a very, very low variant allele fraction, like 0.1% in blood, meaning a super small clone. It's unclear that they had a relapsing polychondritis phenotype. It's unclear how a mutation at that low burden could relate to the phenotype. The paper doesn't directly show that, but it's very interesting. And we've heard of cases where there's X monosomy. So there's, there's only one X, and that happens a lot. Like, you know, hemophilia is an X-linked recessive disease in men, but if there's a problem with the second X and they mutate the one good X gene they have, then a woman could get these diseases. I think that will happen with VEXA, but by and large, this will mostly be a disease confined to men and likely older men.
So I don't know the answer to this question, but you're going to help me. Since it's a monogenic kind of thing, like you say, gene editing would do it. How hard, or how safe, or how risky is gene editing? Because I don't know what's involved in gene editing.
Ideally, it would be more safe than allogenic bone marrow transplantation. So the idea would be that you'd give them agents to stimulate those stem cells out of the marrow into the periphery where you would collect them, and then you can, in the lab, genetically alter them, fix them, give them back to the patient. So in theory, it's safer because you're not having to give, you know, a matched donor and do all of these other myeloablative procedures. You know, it would be one hope towards safer forms of bone marrow replacement therapy.
Well, thank you so much, both Dr. Peter Grayson, Dr. Peter Merkel. Again, a brilliant talk. I learned so much from you today. Thank you so very much. And thank you, audience, for audience participation. It's been a brilliant talk today, and I hope everyone have a wonderful evening. Good night, everyone. See you guys at the next meeting. Bye. Thank you, AV people. Thank you.